Welcome everybody to the roundtable today discussion about regular acceptance of MPS systems. So we are happy that more and more people are joining. The doors won't be closed. Please come in after the very fantastic party last night. And we are hoping that to have an engaged discussion, a lively engaged discussion with you afterwards. So please stand up and ask your questions. So today we will try to look at the term regulatory acceptance from different angles to demystify also a little bit this term and look at this from the perspectives of the regulators, of course, but then also from the users like pharma companies, cosmetics, chemistry, chemical industry, agrochemicals, food industry. So is this, are there different approaches? Is such an approach, is it the same approach for all these different areas? So on the other hand, you know, looking into the submissions to the regulators, what could be obstacles? Why are not so many submissions currently getting through and more MPS systems are used in this context? So we will kick off the discussion with an example of a MPS system, which is the skin in vitro model, which is a regulatory accepted MPS model for a skin irritation assay in replacement of in vivo as an example, and to highlight the journey of such a regulatory acceptance. But before we go into this detail, I would like to welcome also the panelists, and I would like to ask them to briefly introduce themselves with their name, affiliation, and what is your connection to MPS. So my name is Stefan Kustermann. I'm from Hoffmann La Roche, and next to me as my co-chair is Donna.
I am Donna Mendrick from NCTR FDA, and along with Susie Fitzpatrick, we co-chair FDA's Alternative Method Working Group.
So is this on? I guess not. Yeah, okay, Susie Fitzpatrick, FDA, as Donna said, co-chair of the Alternative Methods Work Group, which is all of FDA.
Jean-Loup Dorn from European Food Safety Authority. Connections with MPS is the fact that EFSA is working to implement the use of new approach methodologies in risk assessment. Thank you.
Hello, good morning. My name is Anne Gourmelon. I work for the OECD and more specifically the test guidelines program. And my connection to MPS is that we are trying to evolve our products, our practices to uptake innovative methods in standards and guidelines and frameworks.
Good morning, everybody. My name is Maurice Whelan. I work at the European Commission's Joint Research Centre in Italy. I'm head of the Chemical Safety and Alternative Methods Unit, and I'm also head of the EU Reference Laboratory for Alternatives Animal Testing, better known as ECFAM. And our role is really, we're part of, let's say, the risk management and policy part of the European Union. And we offer scientific advice and support to risk managers and to policy makers, and in particular, we're promoting new approaches to risk assessment.
My name is Seiichi Ishida from National Institute of Health Sciences in Japan and also the Sojo University. And so my scientific interest is the hepatotoxicity and also the regulatory sciences. And I'm being involved in the Japanese MPS project, and I'm organizing the standardization program. Thank you.
So as next, I would like to ask Maurice to start the presentation. I don't know, can you pull up the slides from behind?
Okay, great. Well, my job is just to kick the proceedings off by telling you a story about the very first MPS to gain international regulatory acceptance for toxicity testing, the reconstructed human epidermis. Next slide, please. Oh, I have to do it. So this is a reconstructed human epidermis, close-up of it, basically made from primary human keratinocytes. differentiated into 3 layers of the epidermis, the basal layer, the spinous layer, and the granule layer. And then on top of that, you've got that intercellular lamella made up of, the lipids you find in the skin. So, a real work of science and a work of art, really. So this has become, a key component, a model, the test system in a number of toxicological testing methods. It started out looking at skin corrosion, then developed into a method for irritation as well, for phototoxicity, and then more recently for testing chemicals for their potential to cause skin allergy or sensitization and genotoxicity. Now, they've also, these methods have become international test guidelines at the OECD. So for those you don't know, like what's an OECD test guideline, why it's important? Well, there are 38 member countries in the OECD, and an OECD test guideline is really an agreement between these countries on these methods as being standard methods that underpin the mutual acceptance of data. So if you produce toxicological property data in one member country, it'll be automatically accepted in another country. It also forms the base of being able to carry out these tests under GLP, which is very important for regulatory needs. So test guidelines are really really the highest bar, you'd say, in international acceptance. But what we also have emerged too was performance standards to basically speed up validation of methods which are what we call me too or similar. Because in TG 439, the one on skin irritation, for example, there are actually 7 different methods, 7 different models, 7 different commercial suppliers of those models, all incorporated into the same guideline. So all these methods produce the same information, but in different ways. These then have been elaborated, particularly the skin corrosion and irritation have been brought into testing strategies in IATA so that really you can do a sequential testing and assessment approach for full characterization of your compound, and particularly to help you, for example, decide what class of the GHS your compound would fall into. And then what was important too in more recent years is to develop what we call a good licensing practice, and this guidance document 298 really describes how we can get that right balance between providing accessibility to all these guidelines in different countries by different users, but maintaining the possibility, of course, that there's a commercial activity to provide the models and the methods and so on. As was mentioned, this was kind of groundbreaking because it was really the first replacement method to corrosion irritation for a 3 R's method. And there was, really strong societal driver. This story started, during the 90s, late 90s, early 2000s. And that was even before in the EU, for example, we had the marketing ban on cosmetics that have ingredients tested on animals, which really kicked in 2013. Validation was a central element to all of this, and really the driver in Europe in particular was to valorize the output of research projects. So there was a lot of research money being invested in developing these models, both privately in industry, but also through European funded research programs. This was significant commitment from multiple parties, and it also involved a lot of international collaboration, particularly between partners of the International Cooperation Alternative Testing Methods, of which we're a part. One very important aspect during that whole validation period was ensuring consistency in production of these models. And that involved agreement on particular technical scientific criteria that producers could really use to ensure consistency in batches, consistency across production sites, and consistency internationally between countries developing these models. And that led also to allowing to apply quality systems like good manufacturing practice. All of this also was a strong driver of innovation. So as more validation and standardization was being carried out, this was also helping drive the innovation of the model itself. So over the years, we've seen other functionality, let's say, added to these models, which includes, the metabolic competence, immune response, full thickness models have appeared now, much more high content readouts like transcriptomics and so on. So we've seen this continual evolution of the model itself. So here's some take-home message, I suppose. First was that, look, validation was the key driver behind optimization, standardization, building confidence, gaining acceptance ultimately, and very much driving the MPS model development, both from the, let's say, the scientific sophistication of the models, but also in being able to produce, reproducible, reliable models. I mentioned the importance of this emergence of performance standards. So because with particularly there was a number of different producers, a number of different models, all wanting to measure skin irritation, it meant that we needed to find a way of being able to allow this variety of systems and methods to coexist, but all conform to the same standards. And I think that's something potentially that as we move forward will be an important aspect to embrace the diversity, but equally ensure confidence and so on. The other thing too is, look, it did involve major investment from public and private sectors working together. You don't do this on a Friday afternoon. It required significant concerted effort and investment over time, but the dividends are high. And finally, I'd say, it was an incremental process that picked up pace as more confidence was built. So as I said, we started, with the first kind of output of research results in 98, 99, the late 90s, and then that triggered the validation. First validation study was skin corrosion, which led into the skin irritation validation. And that led in then to other methods being validated against the performance standards, and those guidelines have actually been updated 4 or 5 times and with no better over a decade of time. So as the science and technology moved forward, these guidelines then were then updated to incorporate these developments. So I'll stop there. And I just wanted to mention, if you want to read more about our validation work in particular and what we're doing, developing and promoting non-animal approaches, we've just published our 2021 annual report. I have a few copies here if anybody's interested, but you can download it from our website. And the other thing is we also have a tracking system for getting information on methods which are, undergoing some evaluation or validation towards regulatory acceptance in very different contexts. And it's called TSAR, T-S-A-R. You'll find details in the report. And you can have access to all of the historic validation reports and independent peer review opinions and so on of these methods. So thank you very much.
Thank you, Maurice. So one of the topics that Stefan had introduced very early on, and we want to make sure is clear in this meeting, is different product types have different regulatory backgrounds. So for example, in FDA, there are obviously multiple regulatory centers, each work on their own regulatory laws. So there is a difference between trying to look at an alternative method, for example, from foods, from drugs, from chemicals, et cetera. So I think I'd like to ask Susie to start off the question, which is, how are the approaches similar and different when you go across product types?
Is this on? Okay. How is what similar, different?
What's similar and different between different product types?
Well, obviously, foods is an area that touches you every day, every American or everyone in the world every day. And so food safety is an important area for us. And because we have not very much regulatory authority over it, looking at new approach methods is very important for us for chemicals. They're really for chemicals. And that's why Jean-Luc and I are both part of what we're putting together an international collaboration on looking at introducing NAMs to the chemical sector, which I'll make a little commercial at the end of this meeting for everyone expanding themselves out of the drug box and looking at the broader picture of chemicals. So I think we need them, I think, more than anyone, and we're hopefully working internationally to achieve that. You might want to add something.
Yeah, maybe just something to add regarding chemicals that don't belong to anybody, to industry, contaminants, nobody is necessarily dealing with them. So maybe these methodologies are very useful. We don't have any in vivo data. We may be able to compare with historical data on related contaminants, but maybe comparative studies might be useful in this context.
Any other opinions?
So the big difference between the chemicals and the pharmaceutical is that we have the human data in pharmaceutical field, but we don't have the, we only have the basically animal data in chemicals. So we need to think about what is the standard for each field.
Yeah, and yeah, another difference, I guess, between pharma and chemicals in general, be they pesticides or biocides or cosmetics or industrial chemicals, regarding we are talking about the regulatory acceptance. I guess for the pharma sector, you can accept high cost of MPS, and that goes with the innovation, right? But in the chemical safety testing, I think that can be a barrier to the uptake of innovation, and that's also one reason why it's taking more time to reach the regulatory acceptance until you have innovation that has a business model that is sustainable for testing a large number of chemicals.
I suppose I can add to that. I mean, differences. One is what chemical entities are actually subject to regulation. I mean, there are hundreds of thousands of chemicals in circulation, but only a very small amount of those are actually subject to regulatory oversight. That's the first thing to take into account. Secondly is, depending on the sector, there are different information requirements. In some sectors, there are actually no information requirements. Agencies have to make do with whatever data they can get their hands on, but not necessarily from a registrant. Whereas in sectors like pesticides or industrial chemicals, there's a requirement for companies, registrants to provide certain sets of data. And that amount of information can vary depending on different factors. In Europe, for example, for industrial chemicals, it depends on the tonnage of the chemical that's produced or imported. Bigger volumes of chemicals in circulation, more information is required. In pesticides, you have a complete package of data that's a requirement for assessment, whereas in cosmetics, it's only certain types of ingredients like hair dye or UV filters that are subject to scrutiny. Otherwise, the companies are trusted to carry out a safety assessment internally. So the other thing that varies significantly, and I think people don't realize this, is the actual assessment processes vary between sectors and agencies. So if you're talking about industrial chemical, like Anne mentioned, where money is tight, then it's much more of what some people call a tick box, you know, a standard information requirement that's satisfied with a standard method, and trying to make the whole process as efficient as we can. Whereas if you're talking about a pesticide active ingredient, they are much more sophisticated assessment processes where you've got a lot of experts sitting around for a long time discussing all kinds of data, whether not only the standard required data, but also any data taken from the scientific literature. And the final thing I'd say is don't forget societal concern. There are certain toxicological endpoints that we're as a society more concerned about, like cancer, for example. And also we have sensitivities when it comes to you know, the type of information, how it's generated, depending on the product we use. And we all know we have a cosmetics ban in Europe, and but we don't have it in other sectors, because that reflects societal concern, and we shouldn't be dismissive of that either. You know? So thanks.
Thanks. So I'm hearing that first, the different areas, they desperately need the MPS models because this is a good way to sort out a lot of things, but then there's also different regulations in the different areas. So getting back to the example Maurice has presented, I'm still wondering, being of course very biased from a pharmaceutical company perspective, I'm still wondering, is that Is that then in the end the way we should always aim for? I mean, or are there other ways, I mean, to gain regulatory acceptance? Do we always need to go the full way until having that qualified and validated the systems, or are there also case-by-case examples? And could you think of them to ease also the way for the developers and the applicants to submit their data to the regulators? I don't know who wants to comment on that.
So, I mean, at FDA, people are very interested in seeing new types of data. And a lot of applicants do come in, from my understanding, talk with Cedar colleagues and others, they do come in with new types of data. The question is, when do you want the FDA or even yourselves to take regulatory action based on that data? And are you trying to replace an assay? So the barrier is different depending on what your purpose is. You know, again, if you're trying to replace an animal study, obviously the barrier to acceptance for everybody is not just the FDA, but your colleagues within your companies and everybody is going to be much higher than coming in with an assay saying, here's a supplemental piece of information. And again, people do that all the time. So you don't have to wait till regulatory acceptance to start sending in new types of data. You know, in fact, you can validate an assay within a company and be specific to the company. It's just usually takes a lot of work, so it tends to be more consortium based a lot of times. But again, don't think you have to wait until there's some gold stamp that says now it's validated, now I'm safe to come in, because that's not how this is going to work. It's not how it's worked with other types of new technologies. You know, people start adding data slowly. You know, for example, to try and explain away a problem they've seen, might use the supplemental information. Again, looking at efficacy doesn't usually have as high a bar as safety. So all different ways that you can start submitting data to regulatory agencies and get everyone comfortable, getting your own companies comfortable submitting data and getting the regulators comfortable looking at it.
Yeah, to add maybe to that, in the chemical sector, I guess regarding standards, we stopped a bit talking about replacement and cancellation or ban of animal testing, because we realized that innovative methods, as they become new standards, new guidelines, they can continue to coexist with animal testing, and it's not really a problem as long as the innovative method has well-defined applicability, and the regulators know exactly what they can do with it, what they cannot do, what are the limitations, and then it's fine. And so replacement will come, but we need to allow pioneer methods to come through, to become the new standards, and little by little, we expect that, of course, animal testing will become obsolete.
I might add to that. Well, yeah, first of all, I heard there was a lot of emphasis in this meeting about the replacement of animal testing and animal. Which, I mean, obviously, I'm head of ECFAM, and we get our mandate from the European Directive on Protecting Animals Used for Scientific Purposes. So don't get me wrong, that's a major policy. But there are major other drivers as well that we should be talking a lot more about. First is, as I said, addressing disease burden in our societies. I mean, we have hundreds of billions being invested in Europe at the moment to beat cancer. That's our Beating Cancer plan. And we all know in certain contexts, chemicals contribute to the burden of cancer, but only a fraction of the chemicals in circulation in the EU are actually assessed in any way for carcinogenicity. Why? Well, because it's too expensive, takes too long. It's an economic barrier as much as it's a scientific barrier. The other thing is, we want our industries to do well. We were hammered after COVID, and now we're, heading into another economic crisis caused by a terrible war. So, we're all looking to find ways of making our economies efficient and effective and competitive and facilitate trade. And the other thing is, we want to move into an era of safe and sustainable by design. We don't want to be putting carcinogens and skin sensitizers into consumer products. And we're doing that a lot at the moment. So I think we need to, and of course, we want to move away from animal testing as well. So I think that's the important to, when we think about acceptance and what does acceptance mean, It's way more than purely showing the scientific validity of something. It's fitting it in within a whole, a value added proposition, But I would say about in terms of acceptance, I mean, I always struggle to find language to describe this, but I'm going to give it a shot. What I described there was what we call universal acceptance, and it was acceptance of methods, not data. It's acceptance of methods. The bar was so high because at that level of acceptance, those test methods can be used in multiple sectors, multiple countries, multiple uses, used by multiple agencies. It's the highest. That's it. It's a gold standard. Not because of the method itself per se. It's golden because it is a universally accepted standard. Okay. But then you have acceptance in specific contexts of use. And that's why often I think you see this contrast between, in the pharma, it's all context of use. Of course it is. But in the other sectors. Now, keep in mind, pharmaceutical sector rely heavily on OECD test guidelines, particularly for their preclinical approval. So, I think as well, there's this artificial separation happening there. I'd have to push back on Donna and say, look, validation is an intrinsic part of the scientific process. For you to yourself to believe your own results, you carry out validation. I hope you do anyway. To convince the colleague down the corridor, you do validation. To convince your peers at this conference, you're doing some kind of. So it is an intrinsic part of the scientific process. So I would say to you that the same principles that were followed in those validation studies are the very same principles that a pharmaceutical company making an IND submission or whatever to a regulator on a very case-specific, context of use is showing qualification data, is showing validation data. It's just done within in situ, confidentially, and it's only good for that particular context. So, scientifically, let's say there's no difference. Everybody's still having to do validation and qualification. Just the difference is that universality of acceptance then. Okay, hope that helps.
So let me add for chemicals, a lot of which don't have an owner to mention, and a lot of which are in food or water that you're exposed to every day and your children are exposed to. It's not acceptable if we already know in advance. We know that animal studies aren't the right tools for us. Not saying they don't give good data, but saying it's not acceptable if we see something in your children's food and we say, as a regulator, we'll be back to you in a couple of years and tell you whether that's good or not, because we have to do an animal study. So we're looking at a lot of mechanisms to work with the industry to help us get new tools. We created a public-private partnership with which the food industry, the Humane Society and others are there to talk about what new approaches we can use for chemicals. We're talking internationally with it. Donna and I have a seminar series through the Alternative Methods Work Group. We invite people to come in and present your new methods, and sometimes we adopt one. We've adopted one. I know our agency has adopted one to look at a better way to look at ADME. So I think there's a lot of ways to work with the agency. Our agency, I know you can work with the other agencies through Talks 21 to work on this together as more of a partnership now than it ever was before to bring these new methods. Because we, as a regulator myself, I recognize I need new tools to do my job better, and that's what we're going to encourage everyone here to do.
Yeah, maybe Stefan started the discussion on the fact that we need to demystify what is regulatory acceptance. The thing is, regulators are two communities, and it's first the risk assessors who do the assessment of the chemical and the risk managers who write the way it should be done. The issue also is the fact that it's set in stone that we need animal studies. So in order to change this legislation, maybe what we want to do is to start simple and, let's say, to be practical, we have the hazard identification, we have the hazard characterization, we have a list of endpoints that are set in stone for genotoxicity, as an example that Maurice just gave. We have liver toxicity or whatever. We could look across these different endpoints and compare them with the traditional way they've been set, let's say for genotox, AIMS test or other genotox tests, use MPS and compare for chemicals for which we have the data across for a range of endpoints. Then then the most difficult question would be to derive a safe level using MPS. So hazard characterization, dose response modeling for a particular chemical. But again, we can compare that with historical data. And maybe starting simple like this could lead to demonstrating that we can use such data sets and see how far they are from the traditional ways of doing it. Thank you.
And really, animal tests are, for a regulator, just a tool to get questions answered. And so if we look at the questions we want to answer from those and say, do I have a new way to answer that question? And for this product that I'm looking at, do I really need that question answered? It depending on how you're using it, you know, and how much confidence I need in the answer. If I'm looking at it for priority setting, I can take a lot more uncertainty than I can for a pivotal study, but it's really just answering questions. All the animal studies are, in my opinion, and we need to look back at what we want answered, what we thought it answered with that study, and can we answer it a different way, you know, as good or better than the way we're using it now.
Maybe just a couple of last two things.
Yeah.
What we want also to do is to structure the way we put all the evidence together using this so-called weight of evidence approach. And doing these comparative studies allows us to do that for all the different lines of evidence.
Right, that's right.
Okay, so I'm hearing there's multiple ways. It's not always the way of the skin irritation model we have to go to gain regulatory acceptance. So overall, I would say with that multitude of ways to get to the regulators, so where is then the gap? I mean, are you getting already enough submissions with regard to MPS model systems? Are you happy with that, or do you see certain obstacles which could impede this process? And what could these be?
I'd suggest to you what the issue is, and let's just stop talking about animal testing for a moment and go back to basics, which is most of the reasons why MPS or MPS per se will not be regulatory accepted. It's the methods that will be proposed or the data derived from it would be subject to acceptance or not. But the first thing is lack of purpose. I've heard the last couple of days, fit for purpose, yeah, fit for purpose. Very few developers actually are able to define the purpose of what they're doing. It's wonderful research, but it has no, from a regulatory perspective, it has no utility. So for example, we don't regulate chemicals in Europe based on liver toxicity. Information required, it doesn't exist. Liver toxicity is important, of course, but we don't. So there's the first problem is how in the name of God can we even talk about regulatory acceptance if we can't evaluate fitness for purpose because the purpose isn't clear, okay? The second thing is understanding the decision-making context. Vast majority of MPS developers who are claiming that, you know, want to have their method or MPS-based system considered for regulatory acceptance have no idea about how decisions are made in a regulatory context and make no effort to find out. Thankfully, you all came this morning, so that might show you're all forgetting. But seriously, how can you engage with the community and seek acceptance when you make no effort to understand all let's say the relative complexity of regulatory decision making? By the way, all the information is available, et cetera, et cetera. The next is once you get to that level, so you think you have got your purpose right, you think you actually understand, you know, generally speaking, how decisions are made. Then it's a good time to start the engagement and have a discussion. Because at that point, you begin to realize that regulators in the regulatory community, and I include myself there, don't have all the answers. Because what we're proposing to do is use a completely different technology, new types of methods, new types of information to make decisions where we've been using completely different type of information. So asking a regulator, what do you want? Tell me what you want. It indicates to me somebody who hasn't made the effort to try to begin to understand. Because then at that point, you start the co-design, co-creation process. Because the first step in that is the co-formulation of the problem. How can the decision-making community that's using the information from the community that's generating the information, how can we formulate the problem in a new way that will allow us to make progress? And that's why I keep saying about the animal testing, even though we want to move away from animal testing, of course, is that, oh my God, how many people who are developing new technologies, including MPS, are trying to replace a 90-day rodent study? they're formulating the problem as we need to replace a 90-day rodent study. That's the wrong formulation. And already you can see that we run into a problem in terms of how anything could be accepted.
So regarding to your comments, so I have a question that which sector prepare the experience of using the new method? So it's the industry side prepare the basement of the data for the method assessment, or the regulator also get the some result to get the feeling of the new method. How do you think? What do you think?
Yes, me.
I guess this was now also the invite to the audience, so to engage with you. What are your questions? Come up and ask. Don't be shy.
Can I add something? I mean, sometimes as a regulator, we go out and look for new methods. So we had thought the neuromuscular junction on a chip would be good for Botox testing, and we at FDA felt we had a way to qualify it, and we went to Dan Tagley to give us assistance on it, and he's just announcing a program to work with him and with the FDA for a chip that we think will replace a very bad test. So we're open when we see needs that we have, we're open to looking for other ways to do it too, through our partnerships with EFSA or PPP or other things. So it's not always you're coming to us. We're sometimes going out and, well, we started the whole chip program that way by saying, hey, we do need this. And we're going to work with NCAS to try to develop what's here today.
I was talking with Laura Suter-Dick earlier, and we mentioned the fact that a lot of the people working in this technology originally were working in toxicogenomics, which again reminded me that, there's an excellent book called Crossing the Chasm, which was written originally for the computer industry, but it talks about these early adopters and these people that are interested in new technologies. And there's this big gap when you try to move to the majority of people. So don't make the mistake a lot of us made in toxicogenomics, where you're talking among yourselves with early adopters in your company, and you're not talking to the basic regulators, because you're not going to get your company to move in this direction if you can't get the heart and soul of the regulators and the more conservative people in your company bought into these technologies. It'll just stop for quite a while. Yes, go ahead.
Hi, Katie Conley, Humane Society of the United States. I'm just curious whether the regulators play a role in incentivizing the companies to get the data in front of you. I'm here, definitely hearing that we accept it, submit it to us. How can we incentivize? Is there a way to do a shorter time frame for getting through the approval process or something like that would incentivize?
I mean, I can come a little bit on that. I mean, I just want to be also clear. I hope it's a debate, isn't it? So I'm throwing things out there. Is having the opportunity to submit is not the same. I'm afraid, Susie, of saying that somehow then it's implicitly accepted.
Right.
We have so in Europe we have very strong incentives, legal ones for companies.
Right.
In fact, it's a legal obligation to use an alternative approach if available. But for example, under REACH, there's a legal provision very clearly under Annex 11. It's where you can what we call, I'm sorry, I'm using lots of regulatory speak now, but adapting information requirements. So you can provide alternative information generated with non-animal approaches if it's considered adequate to satisfy the information requirement. But the vast majority of those submissions are rejected by the agency based on lack of scientific credibility, lack of documentation, lack of whole lots of things. So there's an example where we have a full legal framework. You can submit away all the weird, wonderful data, but very little of it is actually accepted. And I'd be curious to know in other agencies, particularly in the US, is that really the case? because I wonder, the problem is in the you know, it's not my domain specifically, but in the pharmaceutical sector, when you're talking about companies submissions, it's a confidential process. We don't know what's being submitted, and we don't know what we hear some success stories that was announced yesterday, but they're you know, they're flash in the pans. You know, it's very hard to get a sense of that. But I'm glad Donna mentioned toxicodynamics. Like that technology has been, it's almost old, you know. There hasn't been one out of 27,000 chemicals registered in Europe, like 3 have omics data. And I know for a fact that omics data was actually never used in the regulatory decision making. It was kind of just sprinkled on top. So incentivize, I mean, you know, even with legal incentives, it's still difficult to really make progress there, I think.
Thank you.
My name is Nordine Ashamaki from Michigan State University. Thank you for the discussion. You covered drugs and chemicals, cosmetics. What about the food additives? How they are regulated, and will organ on a chip have a role there, given the fact we discover more about the microbiota and its interaction with different organ systems and up to the brain, and the influence of those food additives will become more important in the future. Thank you.
So your question is related to food additives and how they're regulated, correct?
Yeah, and would organ on a chip have a role there?
Yeah. So in terms of legislation in Europe, we have a guidance document which basically sets the scene for what type of studies are necessary. The first one is the food additive absorbed or not by the intestine? And then, of course, if it is, then you have the whole battery of in vivo data set. You can imagine that with the MPS, we could first look at absorption, and that's something we've been discussing a lot with Susie. And then if, let's say, it's absorbed, then we look at intestinal metabolism versus liver metabolism and so on, comparatively between rat and humans, but also the impact of the microbiome, if it's hydrolyzed or reduced or oxidized and so on, and potentially the impact it may have on the microbiome at certain levels of exposure. So potentially these MPS could be very useful in looking at these aspects. I don't know if I've answered your question, but I tried.
So in the US, most things come in through GRAS, food additives, so we don't get any data. Packaging, but you know, we already have things we're looking at MPS for mixtures. Food is a mixture. There's chemicals are a mixture. There's no good way to look at mixtures now. Can we use MPS on that developmental neuro, developmental immuno? There's no good animal studies for that. Those are important areas for anybody. Those are areas so we're out looking. We're not sitting passively as a regulator waiting for things to come in. We are out looking for technologies. We have a plan we're working on with EFSA for NAM. So we're looking for things that can help us regulate our products better. And you know, maybe we need a better job of saying what our regulatory questions are to help guide the industry. And that's something we've talked about. But we're not, I don't think anybody in the FDA is sitting back waiting for things to come to them. Do you think, Donna? No, we want these new tools. We have invested a lot in MPS, starting at the very beginning and working with Dan to look at this technology to help us out. So this is something of all the technologies that we've really invested in our time, a lot of time in across the agency.
And just one very last thing. We have a lot of historical data on food additive toxicity. So, and we have QSAR models that can help us to prioritize the way we're going to, you know, we can use this historical data combined with QSAR predictions before we run the MPS experiments to answer specific questions.
Yeah, it would be great to be updated because, I mean, even absorbed or not absorbed, but it still will have an influence as we know today, like we know more than we knew before when this like division has been put. Thank you so much.
I would add that all of the centers at FDA, the researchers are using different MPS platforms. They're using commercial ones. In some cases, they're developing their own. Each center is doing research on MPS. So again, we're not just waiting. We're actually getting involved ourselves.
Hi, I know this is too early question, but I have a question from the viewpoint of validation. To confirm MPS is very reliable or not, we have to use so many positive control and negative control compounds with already known mechanisms. And you mentioned about the regulation for food, drug, and chemicals are different. In that case, number and the variety of the compounds might be different or not.
There's, validation is science in itself, and there is extensive publications by many people in the room here. I've edited a book on validation of alternative methods, and in there you'll find a whole body of knowledge answering those kind of questions about what it would be ideal design of validation study, including chemical selection and so on. So,
it depends on the purpose of the MPS.
Well, again, if you look into the, proper approaches to validation, you'll see that one objective of validation is to define what we call the applicability domain. So not all chemicals can be tested in all test systems in all contexts. So often the physiochemical properties of the chemical that you're testing dictate whether it would be suitable. Again, if we talk from a mechanistic perspective, I mean, if you have a well-characterized MPS that what pathways, let's say, are expressed, then the chemical selection then will also be informed by the. We've run one of our biggest validation studies currently undergoing at the moment, where we're validating 18 different methods for looking at thyroid disrupting chemicals, and there actually the chemical selection was done with an international group of experts who were expert very much in the mechanistic mode of action understanding of chemicals. So as I say, science validation is a serious business, and I don't understand sometimes why really accomplished MPS developers who are obviously very clever, when they come to actually then address, actually demonstrating the scientific credibility of their MPS and their method, actually look like kindergarten kind of approach, So I'd love that the MPS community take, demonstrating the scientific validity and the utility of what they're doing to a decision maker would be. So plenty of literature out there, plenty of knowledge. ECFAM has been doing it for 30 years, so we've put a lot of, and I mentioned SAR earlier, our massive database. Go there, download a validation study report of the many tens of hundreds of methods we've validated. Look at the scientific opinions, and you'll see there what is important to see in a good validation study. So look, if you're serious about regulatory acceptance or the information from your systems being used in any decision-making context, whether it's within industry, safety assessors, or whether it's in a regulatory assessment, then take qualification and validation seriously.
But the cost for validation is very important to regulatory acceptance.
Well,
right. So that's
there's
why
no point,
I'm so curious about the number of the.
Well, here's I did a little back of an envelope calculation that I did a calculation that out of the European money, I estimated over the last 20 years, we spent €1 billion directly on alternative method development. Okay, €1 billion, between about 80% of that coming from public sources, 20%. Now that's the direct funding, not the indirect. And I estimate that probably about 1.2, 1.5% of that money has been spent on anything that would look like validation. I mean anything. I mean basic, like do your experiment twice type stuff. So I don't understand why people say who are spending, I just looked at a system in there, 150 grand for the couple of pumps and an $800 or whatever. Now, I'm not saying that they're overcharging, What any of you guys can afford that, I think you could afford, investing a bit of, money in, yeah, you know what I mean, showing the reliability and the relevance of what you're doing. Sorry, I'm sounding very agonistic here, but I don't mean.
Yeah, thanks. I support everything Maurice said. And regarding the reference chemicals, I think there's a huge need for curated data sets of reference chemicals that are universally accepted. Not reference chemicals that are picked here and there to validate one technology or another or method, but there's really a need for having globally accepted reference data sets that then any developer can use and come up with their proposed solution. And so there's no need to invest for the developer to invest every time in, defining what is the curated data set of reference chemicals they want to use. And I'm saying that for complex endpoints, this is going to be what is limiting the emergence of innovative approaches is the lack of universally accepted reference chemicals for some of the complex endpoints for which we really need to have solutions in the future. We are starting to see some possible solutions, but yeah.
Otávio Presbrave from Brackman. Taking into account the theme of this roundtable, I'd like to say that me personally, I believe that the MPS is the most powerful tool we have nowadays. But I still have some problems in understanding the validation process of MPS. And especially I think that it's very important for the regulatory agencies to understand, they need to be confident about the powerful of the MPS and the meaning and how the results mean for them to register or not a product. And in terms of validation, I'd like to ask you, what kind of validation should we perform for MPS? Validate the methodology itself or the process? And if we can follow the guideline 34 of OECD for validating an MPS also. Thanks.
Yeah, there's not one size fits all response, I'm afraid, for that. I think the principles of the OECD guidance document 34 about demonstrating relevance and demonstrating reproducibility are universal. It's about the credibility, the scientific credibility of the solution you are proposing, be it an MPS or some other type of combination of in vitro, in silico method. And then it's, of course, the processes, how you perform the validation, you need to adapt depending on the MPS, the complexity of it, and but generally the principles remain universal, and this is not going to change. What may be adapted is, it depends if parts or elements of the MPS have already been validated in some other forms in the past. You use the existing information you have, and building on that, you complement with what needs to be added to create this confidence in the proposed solution. And I think involving the regulators is really important from the onset because it's a process, and the appropriation of the technology, of the innovation by regulators is really key. They are going to come and challenge, and this is normal. They want responses, they want answers to their concern, and only when they feel confident most of the concerns are addressed, then they can accept the innovation.
Can I add to that maybe? Just go back to what Anne said about the chemicals is extremely important. It's a good place to start. It's a key aspect. But there are lots of incredibly valuable byproducts of validation exercises over the year has been these highly curated chemicals lists, but very importantly has all the metadata about the chemical in terms of its properties, modes of action, etc. That can be used. And in fact, over the years, we've published several lists covering all different toxicological endpoints. Again, if you look in our status report, you'll find what we call a list of lists. We've got, I think, 85 lists of reference chemicals for all kinds of toxicological endpoints. And so I suggest please don't reinvent the wheel. Use those for your own purpose. Just to talk, as head of BRACFAM, you should be asking yourself the same question in the sense of what's the best way to validate. I mean, look, validation needs to innovate just as much as the MPS, developments need to innovate too. And so we're always looking for ways of optimizing. Validation needs to be as fit for purpose as your MPS that you're developing. And so I would say to you, the first step is do something intelligent. First of all, you mentioned guidance document 34. Look, again, it's often you hear about this OECD validation. Look, again, I go back to my presentation. The aim of the game there wasn't to do the most costly, time-consuming and expensive validation ever. The aim of the game there was to develop an internationally accepted guideline that you can run under GLP and have mutual acceptance of data between 38 countries. Now, as a stepping stone to reach that highest level, there are expectations about the level of validity you would need to demonstrate, reliability and relevance. And a lot of people focus on the ring trials. Oh God, ring trials are so expensive, and transferability. The reason why that's such an important element at the OECD is not because somebody's trying to break your balls on things. It's because how can you deploy a method in 38 different countries, including Brazil, if you can't demonstrate that the thing can be reproduced between laboratories? So we often get this question like, oh, that's an essential. A second, what are you talking about? That's a requirement because of the nature of a test guideline. Do you know what I mean? It's not the universal requirement for validation. I'm very much a supporter of thinking about companies that have proprietary technology that use it in situ in one testing CRO. How can we discuss establishing the validity of those methods why would you want to do reproducibility between laboratories when there's no intention of and that's where I think performance standards become very important,
Thank you.
Yeah, Brad Calvin of Data Sciences. I just want to jump on the validation bandwagon here. Half my career was spent, or over half my career has been spent in the regulated side of diagnostics. So sitting on assay development teams, running R&D organizations. And so, I know what it takes to bring a regulated test out to market for diagnostics and the statistical rigor that must go through, and that development process from proof of concept to feasibility to development and then to verification validation. I think it's important to understand and sort of get the terminology right, because for most of the presentations that I've seen over the past couple of days, these are proof of concept, right? It's 6 compounds, it's 10 compounds, it's 20 compounds with maybe 2, that are so-called negatives for specificity. That's proof of concept, right? It's not validation. So I think it's probably worthwhile for a lot of the groups to get statisticians. There should be more statisticians here to show what validation really is. So I'd like to see what your comments are. Maurice, maybe.
So I guess I should ask, are you a statistician?
No, I'm not, but I've hung around a lot and I've had them on my development teams.